Good day, everyone. Today, I will enlighten you on the concept of the excitation and contraction quickly of the smooth muscle and its significance in the treatment of hypertension and intestinal colic.
The excitation and contraction of smooth muscles begin with the transmission of an electrical stimuli from the presynaptic neuron. The signal reaches the axon terminal of the presynaptic cell, thus allowing the influx of calcium due to concentration gradient. Upon an increase in intracellular calcium levels, the synaptic vesicle fuses with the plasma membrane and acetylcholine is released into the synaptic cleft. The neurotransmitter then binds with the muscarinic receptor, specifically M3. The activated receptor then interacts with the beta-alpha-gamma heterotrimer, promoting a conformational change, facilitating the release of GDP and binding of GTP. Such exchange promotes the dissociation of the complex from the receptor. The heterotrimer then disassembles into a free alpha and beta gamma subunit, with the free alpha subunit interacting with phospholipase C, promoting the formation of two secondary messengers, diacylglycerol and inositol triphosphate. DIG binds with the calcium channels in the plasma membrane, allowing the influx of calcium, thus increasing intracellular calcium levels. IP3, on the other hand, binds with the receptors of the sarcoplasmic reticulum, inducing the influx of calcium to the cytoplasm. The calcium ions bind with the inactive calmodulin to form the active calcium calmodulin, which then binds to the inactive myosin light chain kinase, forming the active calcium calmodulin myosin light chain kinase. The CCMK phosphorylates the myosin regulatory light chain at the expense of adenosine triphosphate. ATP is split into ADP and inorganic phosphate. The energy derived from this reaction is used to change the cross-bridge orientation from 90 to 45 degrees, causing the filament to interdigitate. The power stroke is initiated when the myosin head binds to actin. The complex then binds with ATP, causing the myosin to release from the active site, therefore returning the conformation of the head from 45 to 90 degrees. This process of cross-bridge formation is known as the latch mechanism, wherein there is prolonged binding of myosin to the active site, and release is promoted only when another molecule of ATP binds with myosin. This mechanism promotes energy conservation observed only in the smooth muscle is due to the fewer ATP produced by the muscle. Relaxation is brought about by the dephosphorylation of the myosin light chain due to the activation of the myosin phosphate and deactivation of the CCMK due to low cytoplasmic calcium.
This patient is experiencing intestinal colic. Let's see how atropine works.
During intestinal spasms, drugs containing atropine may be taken in order to relieve contractions. Atropine, a muscarinic blocker, binds with the M3 receptor, which then inhibits the activation of phospholipase C, thus preventing the formation of diacylglycerol and inositol triphosphate.
This patient is hypertensive. Let's see the effect of calcium blockers.
Hypertension, on the other hand, is caused by the constriction of blood vessels and may be relieved with intake of calcium blockers. The calcium blockers bind with the calcium receptors of the cell, thus preventing the calcium influx and the increase of intracellular calcium. with the low levels of intracellular calcium, vasodilation is now induced.